Let's discuss how to write genotypes with linked genes, unlinked genes, or a mixture of linked and unlinked genes briefly. If it is known that two genes are linked, that is, they're on the same chromosome, such as in the case of purple and vestigial, then the two genes are not separated by any symbol. Homologous chromosomes are separated by a forward slash. And so, if we have a dihybrid individual who is heterozygous for purple plus and purple, and also vestigial plus and vestigial, this is how you would write the genotype. The genes would not be separated by any symbols, and the different chromosomes or the homologs will be separated by a forward slash. Another terminology to keep in mind is that of cis and trans. So if two wild type alleles occur together, or if two dominant alleles occur together, or two mutant alleles occur together, or two recessive alleles occur together, that's called a cis conformation. On the other hand, if you have a mixture of wild type and mutant on one chromosome, or dominant and recessive, or vice versa, mutant and wild type, or recessive and dominant, that is called a trans conformation. What about the situation where you have a mixture of linked genes and unlinked genes? Recall that we use a semicolon to separate genes on different chromosomes. And that's the symbol one would use. For example, purple and vestigial are located on the second chromosome of Drosophila, whereas a gene called yellow, represented by the symbol Y, is located on the X chromosome. Let's say we wanted to write the genotype of an individual who is homozygous recessive for purple, heterozygous for vestigial, and homozygous recessive for yellow. We would write this individual's genotype as follows. They have the mutant recessive allele for purple and the wild type dominant allele vestigial plus on one homolog, while the other homolog must have the mutant recessive allele for purple, since they are homozygous recessive for purple, as well as the mutant recessive allele for vestigial, since they are heterozygote. And that is what chromosome 2 looks like. Then we separate chromosome 2 from chromosome X with a semicolon, and we write the genotype of chromosome X. In this case, they are homozygous recessive for yellow, so they have 2 X's, and we once again use the forward slash to separate the 2 homologs, and they are little y over little y. If there are 2 genes whose linkage status is not known, then we separate them with a dot. So we might write this, or this, next. Let us work with visualizing double crossovers between 2 chromatids or 3 chromatids or 4 chromatids, and writing down the genotypes resulting from these double crossovers. We must keep in mind that crossovers only happen between non-sister chromatids, and in this case sister chromatids have the same color, and therefore these two are sister chromatids, whereas non-sister chromatids have different color. And let's go ahead and carry out crossover between these 2 non-sister chromatids. And here is the first crossover. And there's going to be a double crossover between the same pair of non-sister chromatids. This is what the crossovers are going to look like. And this will result in 4 chromatids that look somewhat like this. So here we have the pair of non-sister chromatids that underwent crossover. And now we can write down the genotypes. This one will still be big A, big B, big D. One of the pair of non-sister chromatids that underwent crossover now will have a recombinant genotype, big A, little b, big D. The second of that pair will have the recombinant genotype, little a, big B, little big B, and little d. And the fourth chromatid will be unchanged, will have the parental genotype, little a, little b, and little d. Next, let us try and visualize 2 crossovers, but that which occur between 2 different pairs of chromatids. And so let's say these 2 non-sister chromatids, they cross over. And so we get something of this sort. However, the yellow segment of the first non-sister chromatid has a further crossover event, has an additional crossover event where the chromatid on the top. And so then you get something of this sort. Right? And we can go ahead and write down the genotypes now. So we will get a big A, big B, little d. This chromosome, the second chromosome will give us a big A, little b, big D. The third chromosome will give us a little a, big B, big D. And the fourth chromatid that never underwent any crossover will give us little a, little b, and little b, little d. And the significance is that in this case, 75% of the gametes are recombinant. And that makes us wonder, is it possible to have 100% recombinant gametes? And indeed, that can happen if you have two crossovers between both the pairs of non-sister chromatids. And this is best shown if one lines up the pairs of non-sister chromatids together, like so. And so we may have a crossover between the first pair of non-sister chromatids, giving us this arrangement, and perhaps there is another crossover between the other pair, giving us this arrangement of chromosomes. And let's write down the genotypes. This chromosome will be big A, little b, little d. The second one is going to be little a, big B, big D. The third one is going to be big A, big B, little d. And the last one is going to be little a, little b, big D. And this is 100% gametes are recombinant. And once again, I'll remind you to keep in mind that although it's possible in a single meiosis to have a 3-strand double crossover or a 4-strand double crossover to give you 75% and 100% recombinant gametes, in reality, if you look in the entire population of gametes, the proportion of recombinant gametes will be pretty low, because these events, double crossover between 3 or 4 chromatids, are unlikely events. Here are some practice problems. One is to practice double crossover between 3 chromatids in meiosis in an big A, little b, big D over little a, big B, little d individual. Draw a 3 chromatid double crossover that would result in a little a, little b, big D gamete. For 4 chromatids in meiosis in an little a, little b, big D over big A, big B, little d individual, draw a 4 chromatid double crossover that would result in little a, little b, little d and big A, little b, big D gametes. Next, let us discuss the idea of mapping distance. Let's say I have two genes, A and B, that are separated on a chromosome by 20 million base pairs. And on the same chromosome, I have another pair of genes, C and D, separated by 4 million base pairs. Now, a crossover occurs when there is a double strand break. That means you break the phosphodiester bond between any two bases in a DNA molecule. So this double strand break can happen anywhere along the length of this chromosome. Moreover, these double strand breaks and crossovers are random events, and therefore the pair that is farther apart, AB, there is a greater chance of there being a crossover between AB than there is between C and D. And what this means is that the number of crossovers is proportional to the distance between linked genes. In this example, since A and B are 5 times farther than C and D, the number of crossovers between A and B is going to be 5 times the number of crossovers between C and D. Furthermore, the number of recombinants, or the fraction of recombinant progeny one would obtain, is also going to be proportional to the number of crossovers. Therefore, the farther apart two genes are, the more likely they're to have crossovers, the more likely they are to produce recombinant gametes. This allows us to define a way of measuring how far apart genes are on the same chromosome, and we say one map unit, abbreviated as MU, and this is also called a centimorgan in honor of Thomas Hunt Morgan, is that distance between genes that produces 1% recombinant gametes, or 1% recombinant progeny in a test cross. Let us practice with an example of computing progeny genotypic proportions using map distance. Two genes, A and B, are separated by 10 map units on the same chromosome, so they are linked. In the test cross, big A, big B over little a, little b cross to the homozygous recessive individual, what proportion of the progeny will be big A, big B over little a, little b? So what proportion of the progeny will be hets for both the traits? Let's write out this cross, and we do have a dihybrid individual who is being test crossed. To a homozygous recessive individual. And this dihybrid individual will produce the following types of gametes. There will be big A, big B, little a, little b, and these are the parental genotypes. Since the two genes are linked and it's clear to us that this individual has one chromosome with big A, big B and another chromosome with little a, little b. The other two genotypes that are possible are big A, little b and little a, big B, and these are the recombinant genotypes. Now, our goal is to determine what proportion of the gametes, and hence the progeny of this test cross, have the genotype big A, big B. Since these two genes are separated by 10 map units, 10% of the progeny will be recombinant, since one map unit leads to 1% recombinant progeny by definition. And that also means that 5% of the progeny will be big A, little b, and the other 5% are going to be little a, big B, since one crossover event will result in one big A, little b and one little a, big B gamete. Since 10% of the progeny are recombinant, that means 90% of the progeny are going to be to have the parental genotype. And what that means is 45% will have the parental genotype big A, big B, and the other 45% will have the parental genotype little a, little b. And this, since the tester individual produces all little a, little b gametes, 45% of the progeny will have the genotype big A, big B, little a, little b. Alternatively, one might say, what is the probability of having progeny of the genotype big A, big B, and little a, little b? And that is equal to, by the product rule, probability of having an AB gamete from the dihybrid individual times the probability of having a little a, little b gamete from the tester individual. And the probability of having a big A, big B gamete is 45%, or 0.45, times the probability of having a little a, little b gamete from the tester is 1, since all the gametes are little a, little b, because this individual is pure breeding. They are homozygous for both the little a and the little b alleles. Therefore, the probability of this progeny, big A, big B over little a, little b, is 0.45, or 45%. Next, here is a problem for you to practice. It's the same two genes AB that are separated by a 10 map units, and the cross is still the same. It's a test cross. However, you have to compute the proportion of the progeny who have the genotype big A little b over little a little b.